Chapter 2 Biological Classification. Since the dawn of civilizations, there has been many attempts to classify living organisms. It has done instinctively, not using criteria that was scientific but born out of a need to use organisms for our own use, for food, shelter and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into tree, shrubs and herbs. He also divided animals into two groups, those which had a red blood and those that did not. In Linnaeus' time, a two-kingdom system of classification with plantae and animal kingdoms was developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organisms into plants and animals was easily done and was to understand, but a large number of organisms did not fall into either category. Hence, the two-kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of cell, mode of nutrition, habitat, method of reproduction, evolutionary relationships, etc. Classification systems for living organisms have since undergone several changes over the time, though plant and animal kingdoms have been a constant under all different systems. The understanding of what groups organisms be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over the time. R.H. Whitaker, 1969, proposed a five-kingdom classification. The kingdom defined by him were named Monera, Protista, Fungi, Plantae and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction and polygenetic relationships. Table 2.1 gives a comparative account of different characteristics of the five kingdoms. The three-domain system has also been proposed that divided the kingdom Monera into two domains, leaving the remaining eukaryotes kingdom in the third domain and thereby a six-kingdom classification. You will learn about this system in details at higher classes. Let us look at this five-kingdom classification to understand the issues and considerations that influence the classification system. Earlier classification system includes bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms and the angiosperms under plants. The character that unified these whole kingdoms was that all the organisms included had a cell wall in their cells. These placed together groups which widely different in other characteristics. It brought together the prokaryotes bacteria and the blue-green algae cyanobacteria with other groups which are eukaryotes. It's also grouped together the unicellular organisms and multicellular ones. Say for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group, fungi, and the autotrophic green plants, though they also show a characteristic difference in their wall composition. The fungi has chitin in their walls while the green plants had a cellulic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera and the unicellular eukaryotic organisms were placed in a kingdom Protista. Kingdom Protista has brought together Chlamydomonas, Chlorella, early placed in a algae within plants and both having cell walls, with Paramecium and Amoeba, which were early placed in the animal kingdom which lack cell wall. It has put together organisms which in early classification were placed in different kingdoms. This happened because the criteria for classification changed. These kind of changes will take place in future to depending on the improvement in our understanding of characteristics and evolutionary relationships. Over time, an attempt has been made to evolve a classification systems which reflects not only the morphological, physiological and reproductive similarities but is also polygenetic, is based on evolutionary relationships. In this chapter, we will study characteristics of kingdom Monera, Protista, fungi of the Whitaker system of classification. The kingdoms Plantae and Animalia, commonly refers to as a plant and animal kingdom, respectively, will be dealt separately in chapters 3 and 4. Now we are going to understand the kingdom Monera. Bacteria are the sole member of the kingdom Monera. They are the most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot spring, desert, snow, and deep ocean where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under 4 categories based on their shape: the spherical coccus, cocci, the rod-shaped bacillus, bacilli, the comma-shaped vibrio, and the spiral spirilla. Third, the bacteria structure is very simple. They are very complex in behavior compared to many other organisms. Bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic. They synthesize their own food from inorganic substrates. They may be photosynthetic autotroph or chemiosynthetic autotroph. The vast majority of bacteria are heterotrophic. They depend on other organisms or on dead organic matter for food. Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty area called halophiles, hot spring thermoacidophiles, and marshy area methanogens. Archaebacteria different from other bacteria in having different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in a gut of several ruminant animals such as cows and buffaloes, as they are responsible for the production of methane biogas from the dung of these animals. Eubacteria. These are thousands of different eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall, and if motile, a flagellum. The cyanobacteria, also referred to as a blue-green algae, have chlorophyll, similar to green plants, and a photosynthetic autotroph. The cyanobacteria are unicellular, colonial or filamentous, freshwater, marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheet. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts. Example: Nostoc and Anabaena. Chemiosynthetic autotroph bacteria oxidize various inorganic substrates, various inorganic substances such as nitrates, nitrites, and ammonia, and use the released energy for the ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legumes, root, etc. Some are pathogens causing damage to human beings, crops, farms, animals, and pests. Bacteria reproduce mainly by fission. Sometimes under unfavorable conditions, they produce spores. They also produce by a sort of sexual reproduction by adapting a primitive type of DNA transfer from one bacterium to the other. The mycoplasma, the organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. Kingdom Protista. All single-celled eukaryotes are placed under Protista, but the boundaries of these kingdoms are not well defined. What may be a photosynthetic protista to one biologist may be a plant to another. In this book, we include chrysophyte, dinoflagellates, eubulenoids, slime molds, and protozoans. Under Protista, members of Protista are primarily aquatic. The kingdom form a link with the other dealings with plant, animals, and fungi. Being eukaryotes, the Protistan cell body contains a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protista reproduce asexually and sexually by process involving cell fusion and zygote formation. Chrysophytes. This group includes diatoms and golden algae desmids. They are found in fresh water as well as in marine environment. They are microscopic and float passively in water currents and called plankton. Most of them are photosynthetic. In diatoms, the cell wall forms two thin overlapping shells which fit together as in a soap box. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have left behind large amount of cell wall deposit in their habitat. This accumulation over billions of years is referred to as diatomeous earth. Being gritty, this soil is used in polishing filtration of oils and syrups. Diatoms are the chief producer in the oceans. Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue or red depending on the main pigment present in their cells. The cell wall has stiff cellulose plates on the outer surface. The most of them have two flagella. One lies longitudinally and other transversely in a furrow between the wall plates. Very often, red dinoflagellates, examples, Gaunecalyx, undergoes such rapid multiplication that make the sea appears red tides. Toxins released by such large numbers may kill other marine animals such as fishes. Euglenoids. Majority of them are freshwater organisms found in the stagnant water. Instead of cell walls, they have a protein-rich layer called pellicle, which make their body flexible. They have two flagella, a short and a long one. They are photosynthetic in the presence of sunlight. When deprived of sunlight, they behave like heterotrophs by predating on the other smaller organisms. Interestingly, the pigments of euglenoids are identical to those present in higher plants. Example, Euglena. Slime molds. Slime molds are saprophytic protista. The body moves along decaying twins and leaving engulfing organic materials. Under suitable conditions, they form an aggregation called plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiate and form fruiting bodies bearing spores at their tips. The spore possess two walls. They are extremely resistant and survive for many years even under adverse conditions. The spores are dispersed by air currents. Protozoans. All protozoans are heterotrophs and live as predators or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Amoeboid protozoan. These organisms live in freshwater, sea, water or moist soil. They move and capture their prey by putting the pseudophodia false feet as in amoeba. Marine forms like silica cells on their surface. Some of them such as ant amoeba are parasitic. Flagellated protozoans. The members of these groups are either free-living or parasitic. They have flagella. The parasitic form causes diseases such as sleeping sickness. Example, trypanosoma. Ciliated protozoans. They are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity gullet that opens to the outside of the cell surface. The coordinated movement of row of cilia causes the water laden with food to be steered into the gullet. Example, paramecium. Sporozoans. These include diverse organisms that have an infectious spores-like stage in their life cycle. The most notorious is Plasmodium, malarial parasite, which causes malaria, a disease which has a staggering effect on human population. Kingdom fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have seen fungi on a moist bread and rotten fruits. The common mushrooms you eat and toadstools are also fungi. White spots seen on mustard leaves are due to parasite fungus. Some unicellular fungi, example yeast, are used to make bread and beer. Other fungi causes diseases in plant and animals. Yeast rust causing leukemia in an important example. Some are the source of good antibiotics. Example, penicillium. Fungi are cosmopolitan and occur in air, water, soil and on animal and plant. They refers to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is to prevent food from going bad due to the bacterial or fungal infections. With the exception of yeast, which are unicellular, fungi are filamentous. Their body consists of long cylindrical thread-like structure called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm. These are called zeosyntic hyphae. Others have septae or cross wall in their hyphae. The cell wall of fungi are composed of chitin and polysaccharides. Most fungi are heterotrophic and absorb soluble organic matter from dead substrates and hence are called saprophytes. Those that depend on living plant and animal are called parasites. They can also live as symbiosins in association with algae as lichen and with roots of higher plant as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, and budding. Asexual reproduction is by spores called conidia or sporangiospores or zoospores, and sexual reproduction is by oospores, ascospores, and basidospores. The various spores are produced in distinct structure called fruiting bodies. The sexual cycle involves the following 3 steps. Fusion of protoplasm between 2 motile or non-motile gametes called plasmogamy. Fusion of 2 nuclei called karyogamy. Meiosis in zygote resulting in haploid spores. When a fungus reproduces sexually, 2 haploid hyphae of compatibly mating types comes together and fuse. In some fungi, the fusion of 2 haploid cells immediately result in haploid cells 2n. However, in other fungi, ascomocytes and basidomocytes, an intervening dikaryotic stage n plus n, 2 nuclei per cell occurs. Such a condition is called dikaryon, and phase is called dikaryophase of fungus. Later, the parental nuclei fuses and the cell becomes diploid. The fungi forms fruiting bodies in which reduction division occurs, leading to formation of haploid spores. The morphology of mycelium, mode of spore formation, and fruiting bodies form the basis of the division of the kingdom into various classes. Phycomycetes. Members of phycomycetes are found in aquatic habitats and on decaying wood, in moist and damp places, or as obligate parasites on plants. The mycelium is acipitate and coincidocytic. Asexual reproduction takes place by zoospores, motile, or by apellonospores, non-motile. These spores are the mycelium is septate and branched. Some members are saprophytic and parasitic, while a large number of them are decomposers or of litter and help in mineral cycling. Some examples are Alternaria, Collectoricum, and Trichoderma. Kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll containing organisms commonly called plants. A few members are partially heterotrophic, such as the insectivorous plants, or parasitic. Bladderwort and Venus flytraps are examples of insectivorous plants, and Cascuta is a parasite. The plant cells have an eukaryotic structure with prominent chloroplast and cell wall mainly made up of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plantae includes algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Life cycle of plants has 2 distinct phases: the diploid, saprophytic, and haploid gametophytic, that alternate with each other. The length of the haploid and diploid phases and whether these phases are free living or dependent on others vary among different groups in plants. This phenomena is called alternating of generation. You will study further details of this kingdom in chapter 3. Kingdom Animalia. This kingdom is characterized by the heterotrophic eukaryotic organisms that are multicellular and their cell lacks cell wall. They directly or indirectly depend on plants for food. They digest their food in an internal cavity and store food reserves as glycogen or fat. Their mode of nutrition is halozoic by ingestion of food. They follow a definite growth pattern and grow into adults that have a definite shape and size. Higher form shows elaborate sensory and neuromotor mechanisms. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Silent features of various phyla are described in chapter 4. Viruses, viroids, prions, and lichens. In these 5 kingdom classification of Whitaker, there is no mention of lichen and some acellular organisms like viruses, viroids, and prions. They are briefly introduced here. All of us who have suffered the ill effect of common cold or flu know that effect viruses can have on us. Even if we do not associate it with our condition, viruses did not found in a place classification since they are not considered truly living. If we understood living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Once they infect a cell, they take over the mechanicry of the host cell to replicate themselves, killing the host. Would you call viruses living or non-living? Viruses means venom or poisonous fluid. Dmitry Ivanovsk Viruses means venom or poisonous fluid. Dmitry Ivanovsky 1892 recognized certain microbes as a causal organisms of the mosaic diseases of tobacco. These were found to be smaller than bacteria because they pass through bacteria-proof filters. M.W. Bajornek 1898 demonstrated that the extract of the infected plants of tobacco could cause infection in healthy plants and named the new pathogen viruses and called the fluid as Contagium vivium fluidium, infectious living fluid. W. M. Stanley 1935 shows that virus could be crystallized and crystal consist largely of proteins. They are inert outside their specific host cells. Viruses are obligate parasites. In addition to protein, viruses also contain genetic material. They could be either RNA or DNA. No viruses contain both RNA and DNA. A virus is a nucleoprotein and the genetic material is infectious. In general, viruses that infect plants have single-stranded RNA, and viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. Bacterial viruses or bacteriophages viruses that infect bacteria are usually double-stranded DNA viruses. The protein coat called caspid made up a small subunit called caspomers protect the nucleic acid. These capsomers are arranged in helical or polyhedral geometric forms. Viruses causes diseases like mumps, smallpox, herpes, and influenza. AIDS in human is also caused by viruses. In plants, the symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, dwarfing, and stunt growth. Viroids. In 1971, T.O. Dinier discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber diseases. It was found to be free RNA. It lacked the protein coat that is found in viruses, hence the name viroid. The RNA of viroids was of low molecular weight. Prions. The modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormal folded protein. The agent was similar in size to viruses. These agents were called prions. Prions. The most notable diseases caused by prions are bovine spongiform encephalopathy. The most notable diseases caused by prions are bovine spongiform encephalopathy. The most notable diseases caused by prions are bovine spongiform encephalopathy, commonly called mad cow diseases in cattle, and its analogous variant, Krojosov diseases (CJD) in humans. Lichens. Lichens are symbiotic association, mutually useful association between algae and fungi. The algae component is known as pycobient and fungal component are mycobient, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi and fungi provide shelter and absorb mineral nutrients and water for its partner. So close is their association that if one saw a lichen in nature, one would be never imagine that they had two different organisms within them. Lichen are very good pollution indicators. They do not grow in polluted areas.